Hi, sorry. My name is Dr. Cara Green, and I'm a postdoc at UW-Madison, as I was introduced. I'd like to thank Rob for inviting me. It was quite last minute, and I'm also very new. This is my first CTC meeting, so please be gentle with me. So I'm going to talk a bit about heterogeneity and dietary protein today. Ooh. Oh, there we go. So we often hear the old adage, you are what you eat, but does that mean anything? And what should we be eating to live longer? So as was introduced by Joy already brilliantly, age-associated diseases have a massive societal cost, and it's also not fun. And so we're looking at whether we can change our diet to reduce the burden of age-associated diseases. Most age-associated diseases are related to glucose dysregulation, which is a big part of my work. I started off in the calorie restriction field. And what we do know is that protein is a major driver of lifespan and metabolic health in mice and also partly in humans. So some really beautiful work done by Solon B.A. and Steve Simpson in Australia showed that these are fun, but they're a bit complicated. But basically it finds that if you look at body weight in mice, there's fat intake here and protein intake here, carbohydrate intake here and protein intake here. And as you can see along the protein axis, this is where we see a change in the colors, which basically means that that's what's important if you look at protein, carbohydrate, and fat. So our focus is on protein. And what you can see here is protein dilution in mice and their lifespan. And if you look at the red line here, they have the biggest increase in lifespan and they are eating the least amount of protein. So we think it's pretty important. And whilst much popular media would suggest that a high protein and low carbohydrate diet is important, that's actually not what the science says. So sorry, but you'll be fine. So low protein diets in humans are actually associated with reduced cancer and mortality, and high protein diets in humans across massive populations are associated with a doubling of diabetes risk and an increase in cardiovascular mortality. That was too quick. We also did a clinical trial where we found that a low protein diet reduced adiposity in males. So this is actually a hamster, not a mouse, but in mice, what we find is that a low protein diet improves a lot of markers of diabetes and obesity risk. So you can see blood glucose is lower, insulin is lower, HOMA2IR is lower. The great thing about protein restriction is that these mice actually eat more calories overall. So if you're picking between CR and PR, I would go PR. And that's always fun. So the first thing we wanted to look at was whether the response to a low protein diet was dependent on sex and strain and the amount of restriction, and were there any conserved changes. So these were my two grad students, Kat and Jessie. They were awesome. And we looked at 3 different strains of mice. So the classic C57 Black 6, which always has a good response to everything. The DBAs, which tend to be a bit more resistant, and the Het3s, which are genetically diverse and a bit more human-like. And we lowered their protein by a third each time. So 21 is control, 14 and 7. And I don't need to tell you actually about the heterogeneous mice, but they're really lovely to work with if anyone hasn't. And what we found was that sex and strain both have an impact on weight change. So you can see the blue line here. The mice on the lowest protein have the lowest weight, and 21 and 14% protein didn't have a big effect. No effect in females. They seem to be resistant to this change. DBA males and females didn't really have an effect, but the DBA females did start to lose weight towards the end. And there was an impact in Het3 males, but not in females. We also looked at glucose homeostasis as very important. We found that there's a significant impact of a low protein diet in males and females. Absolutely nothing in the DBAs. It looks like I've fake drawn these lines, so little changed. And in the Het3 mice, we found males had an impact and females didn't. And this is just looking at the fasting blood glucose. It mirrors what I've shown. How does this happen? Because the mice were eating more, the ones that lost weight. So we look at something called FGF21. FGF21 regulates energy homeostasis and the use of carbohydrates and fats. And what we found, and we also put the mice in metabolic chambers, and what we found was that the C57s had an increase in energy expenditure which was probably accounting for some weight loss. And the other mice didn't really seem to have many changes. This is just all the different things we measure. And what you can also see here is that the C57 males have this massive uptick in circulating FGF21, and it's added to the C57 females, but none of the other groups do. And in the males, we have increased liver expression of FGF21 as well. But it's definitely not a perfect correlation with weight loss and FGF21 expression. And that's pretty interesting because a lot of people have done work in C57s and they say that it's because of FGF21. So we looked at liver transcriptomics, metabolomics, lipidomics, and the phenotypes, and we correlated everything together and did some hierarchical clustering. And what we found, which we kind of expected at that point, was that energy expenditure and FGF21 actually don't co-correlate. So you see, they both correlated with thermogenesis genetic changes, but not together. And what we did find was adiposity actually correlated with this longevity regulating pathway. And we looked at some of the genes in that pathway, and we found that they were all downstream of inhibitors of mTOR, and mTOR is a big lifespan regulating protein complex. You can ask me more about that later, but I don't have time to get into it. But that was really interesting. And this is very busy, but what I'm going to say is if you start protein restriction late in life, it also works pretty well. And this is all published, so you can take a look if you want. So broadly, what we found was that a low protein diet is beneficial. It seems to be beneficial across the board, but there's big sex and diet changes. But more related to this talk is we looked at 3 strains, but as is clear, there are a lot more than 3. And so the next thing we want to see is what is responsible, because we know there's changes in strain, but we don't know what genes are important. And this is a bit more relevant to the CTC. So what is the range of beneficial effects on metabolic health? And are there any key factors or genes responsible for promoting these benefits? And so with Annalise and Grace, my new grad students, we've looked at about 40 BXD strains so far. We put them on a control and a low protein diet. We used the 7% because the 14% didn't really have much of an effect. And we did some metabolic phenotyping. And I'll show you this graph that you've been shown about 8 times today. This is how you get BXD mice. I don't usually present to such a genetically minded audience. And I'm also regretting skipping all my genetics lectures. It's all way over my head. And what we found was that across all these strains, you see this. So they were on the low protein diet for 8 weeks. And what you can see is there is a massive range of response to a low protein diet. So some mice are gaining weight and some mice are losing weight. You can see our founder strains here, so C57 and DBA. And like I said before, the C57s respond well and the DBAs don't respond at all. What was also interesting is we saw a massive sex difference. So if you keep your eyes on BXD13 here, when we look at females, BXD13 is over here. So there is also a big strain effect in these mice. Not all of the, this is just showing strain by strain, the sex difference. And what you can see is they basically never respond the same. We saw a similar response in fat mass and lean mass, and basically all of the other parameters we looked at. What was really interesting is when we did glucose tolerance tests, what we found was quite a different picture in that nearly across the board in all the strains we looked at, mice seem to have low is good for GTTs. You want to lower the area under the curve. And in general, we found that the mice had a positive response to a low protein diet, even if they were potentially gaining weight, which sort of suggests that in some strains, weight loss and glucose tolerance are related. So you can see here, this is a glucose tolerance test. So we inject them with glucose and we watch the glucose go up in their blood. And you can see in the BXD1s, they have a big lowering, and in the BXD11s, it actually makes them glucose intolerant. And a similar story in the females. When we looked at insulin sensitivity, here on the left, we found actually, again, we found a more normal pattern. So it's just that insulin and glucose actually aren't having a massively similar effect, which was pretty interesting. And we looked at FGF21 as well, because this seems to be pretty important. Again, sorry, this one's missing, but what we found was that in some strains, so BXD44, we found this upregulation in the males on a low protein diet and in the BXD48s, but not across the board. So again, the FGF21 story is quite incomplete, but it's still interesting. And when I just correlated all of the phenotypes we looked at, oh, this is adiposity change, it's lost its label, with all the strains, what we find is that, kind of like I said, if you look at glucose tolerance and fasting, there seems to be a positive response with protein intake, so low protein, low fasting blood glucose. But if we look at fat change, it seems to have purple and pink, so a range of responses. And then we did some QTL mapping with Carl, who I don't think is here. Online. He's online. Hi, Carl. To try and identify molecular markers, to try and figure out what's going on and what could be important. And this is just an example of one of the traits. I think it's body weight change. So you can see if you have this D allele, you tend to have an increase in weight. And if you have the BB allele, you tend to have a weight loss. Lean mass actually seemed to give us our strongest marker, which I think is really interesting because one of the big problems we have as protein restriction researchers is sarcopenia and bone weakness with age. And people get very stressed out about low protein diets in elderly people. But in this case, it seems like if you have the BB allele here, you actually don't have lean mass change on a low protein diet. So if you're having glucose improvements, but no lean mass loss, then that's kind of what we're aiming for. And one of our QTLs did reach significant threshold. I'm actually not going to tell you what is here. One, because I'm still adding BXD strains to this analysis. And two, because I'm looking for a job. So if you want to hire me, I will tell you. But it seems to be on chromosome 19. And maybe you know what the gene is. You might be able to guess. And these seem to be sex specific. So we found big changes in the males when we looked at lean change, but we actually found more differences in fat change when we looked at females, which might be more related to the estrogen effect that has the effect that estrogen has on fat. But again, reach significance. And so future directions. So I work a bit with Dr. Brian Parks at UW-Madison. And what we're hoping to do is identify a gene list and then relate this to some human GWAS data that's of relevance and target our gene discovery downwards and hopefully create some knockouts. Ooh, this is what I just said. So just to end, we aren't what we eat. The response is dictated by genetic background and sex. If humans respond the same way as mice, you might not respond positively to a low protein diet. And we would like to look at genes to try and figure out if it is or isn't beneficial. So yeah, thanks to the lab. They're all wonderful. Jessie, Kat, Annalise, and Grace, who help with this work. These guys are in the lab who help a lot. Funders and Dudley, my boss. And yeah, if you have any questions, I'd be happy to take them.